Then genetic toxicology studies. This also needs to be GLP, and you need to do this in prior to phase 1. This also is needed prior to phase 1, safety pharmacology that I mentioned. Genotoxicity studies are also needed prior to phase 1. Why you do this? Your objective is to detect if your drug is going and interacting with the DNA. Is it causing any damage to the DNA? What is the mechanism? If it is doing some interaction with the DNA, what is the mechanism? And also this helps to predict the carcinogenicity risk, whether this genetic manipulation or genetic interaction, DNA interaction, and genotoxicity, will it lead to the cancer, formation of cancer in patients over a longer period of time? If you take the drug for 5 years, 10 years, what happens? And of course the safe clinical development. But this is very, very important aspect. Believe me, with my experience in pharmaceutical industry, any drug that has any concern or potential for genotoxicity, no company will take that forward. The molecule gets dropped there, because no one wants to take a risk that you want to treat a disease, but then that has a potential to cause genotoxicity and a cancer eventually. You would not want to take that risk. So any concern with the genotoxicity, that's not taken forward. Usually, the regulatory agencies require uh, 3 different assays that you need to do. 2 in vitro, this is called EMS test in the bacteria. Another one is in the mammalian cells, and then there is one in vivo study in the animals. The 3 tests that are mandatory. Repeat dose toxicity studies. So this is repeat dose itself tells you that you give the drug or the candidate or the molecule to the animals every day and see their long-term effect, what happens on different organs. So why you do these studies? To identify the target organs. Okay, whether it is affecting liver or heart or kidney, or what is it doing. Characterize the dose response. We talked about this earlier. Establish a NOAL. This is an important concept. No observed adverse effect level. So this is the dose where there is no toxicity in animals. That's very important dose that you consider when you project the human dose. Systemic toxicity you evaluate, you monitor the clinical pathology and biomarkers. Sometimes biomarkers is very important. So you see that your drug is causing a liver toxicity, but there is a biomarker. It changes the liver enzymes before causing the damage. That helps you. When you go to clinical trials, you monitor those enzymes. And if you start seeing some increase in the enzymes, stop the treatment. That's very important. That's called biomarkers. Like you see some toxicity, but is there a way to predict that toxicity? Or is there a way to monitor that toxicity? And that's where biomarkers are very helpful. Evaluate reversibility, recovery. Okay, there is a toxicity, but if you stop the drug treatment, is it going to be reversible or is it a permanent one? That's important aspect. Regulatory submission and clinical monitoring strategies that I talked about. As per the health authorities, you need the studies in 2 different species, one rodent and one non-rodent. Rodent can be mouse or rat, sometimes also hamster, but usually rat or mouse. And non-rodent is dog or mini pig or monkey. One of these species you have to use. And how do you choose whether you want to do it in dog or mini pig or monkey or rat or mouse? Based on the PK profile, and metabolism pattern, which one is most relevant to human? Target receptor homology. So you have a particular target or the receptor. How is that receptor expressed in human versus that animal? That if there is a good match, then I think that is the right species for you. And then also the pharmacological effect, affinities. Then typically, what is the duration of these studies? If for whatever is your clinical trial duration, you need to cover that duration in the toxicity studies. 2 weeks in the clinical trial, you need 2 weeks in rodents and 2 weeks in non-rodents. For clinical trials and for marketing approval, you need higher duration. But it matches only up to 6 months. But beyond 6 months human treatment, you only do 6 months in rodents and 9 months in non-rodents. You don't need to do it beyond that. Even if your drug is intended for 10 years for diabetes treatment or 20 years in human, you still don't have to do it for 10 years in animals. I see a hand raised. There is a question probably. Go ahead, Shidhar. Is there a question?
Hello, sir. There is one question in the chat box.
Okay.
At what stage of the drug discovery do you recommend integrating in silico toxicity tools such as pre-HERG, Protox, and Carcinopred?
Yeah, they are early, very early in the discovery. Before, when you are screening multiple compounds, because this is in silico, it doesn't cost you a lot of money. You don't have to spend a lot of time or use animals. So you do it early on. So if we have compounds, like I have 100 compounds, I can put 100 compounds into the software and it will tell me which compound is best and which one is not good, where there is a concern. Of course, it's a prediction, but it helps us to prioritize which compound is good. So that is used very early in the discovery stage.
Yes, sir.
Then we can again talk about it. Okay.
Yes, sir.
And then you also need to look at the reversibility. Like, say, example, you typical design is you start treatment for 3 months and then you stop the treatment and still follow the animals for another 1 month and see if you are seeing some toxic effects during the treatment. Once you stop the treatment, whether they are reversible or not. This is important in patients because you start giving medicine to the patients and you see some side effects. Doctor may say, okay, stop the drug. But once you stop the drug, will it reverse the toxicity or the side effects? That's an important aspect that we evaluate in these studies. What we evaluate in these repeat dose studies is a huge list, but I'm just giving you a very highlight. We look at the clinical observations, any abnormal behavior of the animal, any distress, etc., body weight, whether they are eating or not, drinking the water or not, hematology, the blood count, clinical biochemistry is a huge panel that we look at all the biomarkers, urine analysis, look at the urine abnormalities. Then there are different organs, we weigh them, we look at their pathology, we do the histopathology evaluation to see at the microscopic level in panel of tissues, 30-40 tissues, whether or not there are any microscopic changes. We do special assessment of eyes, ECG, respiratory function, etc. So this is a huge panel that we look at when we do these toxicity studies. Then there is another type of study that's called a development and reproductive toxicity study. There are important 3 different categories here: segment 1, segment 2, and segment 3. Segment 1 evaluates male and female fertility. When you give your drug to the patients, is it going to cause any damage or any effect, harmful effects on the fertility, on the sperm count, or the fertilization? It's important because someone may be taking this medicine, but then that may have an adverse impact on the fertility aspect. Segment 2 is on the pregnant animals that we do to evaluate what happens if the drug is given to the pregnant woman. This is very, very important. There are a lot of cases where some drugs were given to the pregnant woman and then the fetus got damaged and then the baby was born with some abnormalities. So this is very important aspect that we study. It evaluates embryogenesis, fetal development, etc. And then there is a segment 3 where what we do is we give it to the rats or mice and follow their next generation. So they are treated, and then when their next generation is born, then we follow the next generation as well. That's called pre and postnatal developmental toxicology study. So this is the entire cycle that covers all aspects of the reproductive and developmental toxicity. This also needed as a GLP compliance manner. Carcinogenicity study. So this is the study to evaluate whether your drug can cause a cancer, right? So there are 2 different ways a drug can cause cancer: genotoxic mechanism and non-genotoxic mechanism. Genotoxic mechanism is your drug goes and interacts with the DNA. It breaks the DNA or does some mutation or some manipulation with the DNA, and that leads to the malignant cell because of those mutations, and that can cause genotoxicity, and that can eventually lead to cancer formation. Or non-genotoxic mechanism, where your drug is not interacting with DNA or binding to DNA, but the mechanism of your drug target or drug interaction is such that it leads to proliferation of the cells. Example, epidermal growth factors, right? So it will lead to the increase in the cell proliferation, and that will lead to cancer formation. So it's important that we evaluate this risk. We evaluate the genotoxicity risk early on, but we also do carcinogenicity studies where we evaluate together both. And this is also GLP study, and you need it. This takes very long time. You have to do the study for 2 years in rat and 6 months in transgenic mouse, the genetically manipulated mouse, and look at whether or not those animals are forming the cancer, after the chronic treatment. Someone may ask, in humans, the drug we may take for 10 years or 20 years, but in rat, we were testing only for 2 years. And how do we translate this 2 years to the 20 years in human? But let's keep in mind that the lifespan of the rat is 2 years. So rat will survive for 2.5 years. So when you give a rat a drug for 2 years, it covers its entire lifespan, proportionate to human's lifespan. So whatever you do for the entire lifespan of rat, that can predict what will happen in human with a great accuracy, I would say, if not 100%, what will happen in human if you get that drug for a lifetime. So this is a very, very important study, very expensive, very time consuming. It's a 2-year treatment, and then we do a lot of evaluation. So it takes roughly 3.5 years to complete this study. It's a very expensive study as well. But we don't need it for clinical trials. We need it only before it is approved for marketing. Now, what is the role of regulatory agencies? These are all different regulatory agencies. FDA is in US, then there is a European Medicine Agency in, we call it EMA in Europe, PMDA in Japan. We have a DCGI, Drug Control General of India in India, MHRA in UK, and then every country has got their own regulatory agencies which regulate every step of your drug development and approvals, right? Without approval, you cannot do, legally, you cannot do any clinical trials without approval from health agencies. And we have looked at this, right? So research and then the clinical development, then review and then marketing, and then IND application goes to health authorities, and then the before approval as well it goes, and then you continue to have continuous interaction during the development to seek their guidance. So this is very, very important, and all these agencies develop the guidance that okay, this is what you should do, this is expected, this is a data package that we need, and they also do a pivotal role in guiding you on your development process if you have a doubt, and do all the approvals. And there is also something called ICH, International Council for Harmonization. This is the body that covers FDA, EMA, everything, because every country has a different requirement earlier. So there was an intention that okay, as a company, we should have only one requirement that will be accepted globally, and that's where this ICH was formed, and they develop the guidance documents or the guidelines that everyone has to follow. And if you follow ICH, then your data is accepted in all these countries. All right, so we covered about the different toxicity studies. Now, what if the drug is toxic? Right, this is a very, very important question. Okay, we're doing the toxicity study, and many people say, why take a high dose, take a low dose? Why do you want to see the toxicity? In purpose of the toxicity study is to characterize the toxicity, not to not have a toxicity in the animals. You want to have a toxicity, and then you see at what dose it is causing toxicity, what kind of toxicity it is causing. So Paracelsus is like, is the man who pioneered the use of chemicals in medicines, and he made a very good statement. All drugs are poisons. The benefit depends on the dosage. So any drug that you take is going to have a toxicity for sure. Is the dose that differentiates whether it will have a therapeutic effect or it will have a toxic effect. Say example, you take one pill. Someone has an insomnia, right? Cannot sleep. Doctor prescribes you a sleeping pill. You take one pill, that is for your sleeping, that's a therapeutic effect. But if you take 100 pills, it turns into poison because the dose is high. Same is with those who drink alcohol, probably would know one tequila shot versus more. Or even the x-rays, you take one x-ray is safe, but when you are exposed to the huge radiation, it's toxic. And table here, if you see, it gives you an example, because it's only the dose that differentiates. The botulinum toxin is probably the most potent toxicant known so far, and its LD50 is really 0.00001 milligram per kilogram. Just imagine how potent it is. So at such a low dose, it can cause toxicity, and then you keep on going, look at these drugs, and this is just few examples. And then even at a particular very high dose, vitamin C is also toxic at a particular dose. So it's the dose that differentiates whether it will elicit a therapeutic effect or a toxic effect. Otherwise, every drug is toxic. It's just the dose that differentiates it. We need to remember that. And what are the important considerations for this? Is the toxicity relevant to human? Can we monitor this toxicity? Like in human, like if there are some damage to the liver, can we look at some blood collection and then look at the liver enzymes? Is this toxicity reversible? Very, very important. You see some toxicity, but once you stop the treatment, will that come back to normal or not? Is there a safety margin that, okay, you see toxicity at 100 milligram dose, but you are only giving 2 milligram or 5 milligram for therapeutic effect. So there is a margin. So that's very important. So if your efficacious dose is 10 milligram per kilogram, toxicity is at 100 milligram per kilogram, you have a margin of safety. That's called therapeutic window. That's tenfold. And the risk benefit analysis, I will cover this further in next slide. Very quickly, what if the drug is toxic? What is therapeutic index and safety margin? So This is a graph, say, dose response curve for any drug, sigmoidal curve we all know in the pharmacology. As you increase the dose, your effect keeps on increasing. So this is called NOAEL. Okay, there are different concepts, NOAEL, NOEL. So this is NOEL, sorry, NOEL is the dose where there are absolutely no effects. Absolutely no effects. Then NOAEL, that there are effects, but they are not adverse, they are not toxic. And then there is MTD. This is called maximum tolerated dose. That's a maximum dose that if you increase the dose further, your animal will die or there will be severe toxic effects. So these are important concepts. We usually go with NOEL and NOEL. That's the benchmark for any safety projection. And this is, I already explained, but I think this graph at the bottom is easy to follow. You have a time and then there's a drug level in the blood, okay? And then you have this dose response curve. So at this range, at this concentration or dose, there is no activity, pharmacological activity. This is the dose where pharmacological activity starts. And when it reaches this dose, the toxic effects start. So this window that is highlighted between the minimum dose where it is pharmacologically active and a minimum dose where it is toxic, that's called therapeutic window. And we have to play with this window, make sure that in human we do not cross this level. This is very, very important aspect of risk assessment. And there is something called risk benefit analysis. Every drug you are bound to see some toxic effects. And how do you make a judgment whether or not this is an issue or not? And that's called risk benefit. What is the risk with this drug and what are the benefits you are going to get? And I will explain it with a very simple example. You are developing a drug for treatment of diarrhea, but its side effect is, or toxicity is, it causes cancer. So your benefit is you are treating diarrhea, but your risk is it may cause cancer. Are you willing to take this risk? Will anyone want to take such a medicine to treat diarrhea that can cause cancer? No. That's where it's very high risk and no one will take this drug. But on the contrary, drug for the treatment of cancer that causes diarrhea. So you are taking a drug for cancer. You know chemotherapeutic drugs, they cause a lot of diarrhea in patients, but patients still take it because here you have a risk of diarrhea, but your benefit is much, much bigger than your side effect, than the risk, because you are treating cancer. It's a life-saving drug. So you are treating cancer. I'm okay with getting some diarrhea, some vomiting, lose my hair, but if it's going to treat my cancer, I'm okay to take that medicine. This is called risk benefit analysis, very, very important part of decision making process. What is the biggest challenge? I just have a couple of slides. I will finish it and keep some time for discussion. So biggest challenge is the translation from animals to human, because people always say that animals lie because animal anatomy, physiology is different than human, right? A mouse is 20 grams or 30 grams. Human is 70 kg. How do you translate what you see in mouse? How do you know that it's going to predict what will happen in human? So there are always gaps. There is no model that is 100% predictive in terms of efficacy or the PK or the toxicity. But then you have to human. And when I talk about translation from the cellular level, when you do some assay in the cell, individual cell versus the tissue or the organ, not tissue, I would say organ, say heart or lung or liver, and organ was to human as a whole system, or cell to animals and then animals to humans. So this is a complex integrated translation process from cell to tissues, tissues to organs, organs to human, animal to human, all those things. And it's very different. And then why do you see these gaps in the translation? Because of differences in the genetics, difference in the anatomy and physiology in animals and human. There are a lot of tissues that are present in animals but not present in human, and vice versa. Target biology and toxicity mechanism, the mechanism can be different. Then the PK properties, the pathological state of patients. Very important thing to note. All the toxicology studies are done in healthy animals, normal animals. But when it goes to the patients have a pathological state. They already have a disease, diabetes, cardiovascular, whatever, the cancer, they have a disease. So that pathological state can also aggravate, your toxic effects. Study design and methodological flaws. Sometimes your toxicology studies are not designed properly. That can reduce the prediction power. Lack of human relevant biomarkers. The dose projection in human is not right. I explained to you how this therapeutic window is. If you don't project the right dose, then you lose the translation part. And the safety margins. You don't have enough safety margins. So this is all, and I think nowadays there is no talk or no discussion is complete if you don't talk about artificial intelligence. So I know I just want to present a couple of slides talking about artificial intelligence, how it is helping and changing the way we do the drug discovery and development. It's very important, and this is really, I'm not an expert in artificial intelligence, but then there are people and there are teams which are working on this dedicatedly. But this is a topic in itself for another session. But very high level, I want to mention that the traditional drug discovery process, the pipeline, and then the outcome is we have seen, like, you start from thousands of molecules and you arrive at only one, and then it takes a long time, very intensive, expensive, and you have a less predictive value. How can artificial intelligence help you to have a better prediction, early killing, because sometimes you kill the drug early, you kill it cheap. Rather than investing more money, you kill it early and then you save a lot of money, and it reduces the drug failure ultimately. So if you look at this slide, in general, this is a drug discovery process. We talked on this earlier, the target validation all the way to marketing. And how can artificial intelligence help you? It reduces the development time because you can predict a lot of things with artificial intelligence. It lowers the cost. It reduces time, cost. It enhances the personalized medicine. It increases your success rate in general, right? And if you see here, artificial intelligence can help you. So what people do nowadays is, you have the chemical structures, you put them, and artificial intelligence can already predict a lot of things actually. So it's also part of the in silico that we spoke in the beginning, is getting more and more accurate because of artificial intelligence. And there are a lot more, but there are still some limitations on the data, because essentially what it does is, you have to put some data set. It's artificial intelligence, right? It gives you the input, output based on the inputs that you give. So you have to have a training data set, that all the information that is accurate, you feed that into the system, and then you testing data set, you put some new structures to test how it predicts, and then there is a deep learning and machine learning, etc., and then it gives you the prediction. But the quality of training data set is very important. So how you feed the information is one of the limitation. I think that's improving. And the model, that in silico model that you develop, or the artificial intelligence model that you develop, it should not have any bias, or in the model, and it should have a better predictability and the confidence in general. That's a very, very important aspect. And I think as we make more progress, more data will come out, and I think this will eventually make successful drug development rate higher. It will increase the success and decrease the failure.